Hi, in this video we're going to continue our talk about the urea cycle, specifically how those ammonium molecules get into the hepatocytes mitochondria. Even more specifically, we're going to kind of focus on the cofactor PLP, or pyridoxal phosphate. Okay. If this cofactor sounds familiar, it should. If we go back from earlier in the semester, this is vitamin B6. Okay. So anyway, it's going to be an important cofactor in these enzymes called transaminases. Okay. There are many transaminations, but they all, for each individual amino acid that they transfer the amino groups for. But they all have the same kind of function, okay, using PLP. And pyridoxal phosphate is also called PLP. Now, to go from big to small, when you have your specific amino acid going into that transaminase, it's going to come into contact with the PLP already in what's called an aldimine form. Okay, there's basically that PLP bound to one of the carbons of PLP bound to a amide or a nitrogen. Okay, you have internal aldimines and you have external, depending on whether it's the PLP is bound to the enzyme or it's bound to the amino acid. Okay, so when you have that amino acid coming into that transaminase, with PLP, you could have three different reactions take place. A decarboxylation, where our carbon comes out in the form of CO2. A racemization, where you'll have your L amino acid turn into a D amino acid, or the one we're going to be focusing on, a transamination, okay, where we're going to take the amide group from the amino acid, and we're actually going to eventually give it to alpha-ketoglutarate to turn it into glutamate to be transported into the liver. And that's if it was any cells that are not muscle cells. If it was a muscle cell, there is a specific molecule that's not alpha-ketoglutarate, and that we add that amide group. Okay, try to remember what that is. Okay, for muscles, it's going to be pyruvate into alanine. All right, so that's a good question that he could ask. For cells, it would be alpha-ketoglutarate to glutamate, but for muscles, it'll be pyruvate to alanine. But same thing, they both work in the same ways, okay? You can view pyruvate as an alpha-keto acid, so kind of like the same as alpha-ketoglutarate. They go through the same cycles. And it's going to be that transamination that we're going to be focusing on in this video, okay? Just know that they could also do the decarboxylation or racemization, all right? So, focusing on that transamination, there are basically 4 steps, but in reality there are 8, and we'll get to that, okay? With that L-amino acid coming in, it's going to bind to that aldimide form of PLP, where it's actually going to replace that enzyme, okay? So it's actually going to turn into what's called a Schiff base intermediate, or that other name for external aldimine, with that PLP with the now amino acid, okay? So even though we're inside the enzyme, it's not really taking place for our purposes, okay? At this next step, we are actually going to have we're going to kind of kick off a hydrogen from that amino acid to form a double bond between the between the carbon of the ring of PLP with the external carbon that's bound to the amino acid. Okay, so we'll form a double bond. All right. And this is going to create what's called a quinonoid intermediate. Try saying that 5 times fast. Quinonoid intermediate. Okay. Uh, that quinonoid intermediate, wow, that's a weird word. That quinonoid intermediate is then going to use that double bond between those two C's that we just created to attack a hydrogen, alright? And this is basically just a rearrangement, okay? Um, and that rearrangement can then be hydrolyzed with water to take off the now alpha keto acid. That amino acid is going to leave its amide group with PLP, which is now going to be called the pyridoxamine phosphate. Okay, pyridoxamine phosphate. So now those are the 4 basic steps. But if you're thinking, wait a sec, how does alpha-ketoglutarate get it? Well, it's actually going to go through those same 4 steps in reverse. Okay? It's that alpha-keto acid. All of these steps are reversible. So that amino acid goes in, it's going to take its amide group and leave it with PLP. And then, if it's cells that are not the muscle, alpha-ketoglutarate are going to come in, going to go in reverse and take that amide group. If it was in the muscles, a pyruvate would take the place of alpha-ketoglutarate, and then that pyruvate would turn into alanine. At that point, those glutamates and alanines are going to go into the mitochondria of hepatocytes, and then we'll talk about how they leave off their ammonium to turn into eventually either aspartate or the carbamoyl phosphates. Okay.